Now let us draw the transcription bubble. First of all, I will draw the DNA. The site where the transcription will begin will form the transcription bubble, and as the RNA will be formed, this whole transcription bubble will move forward till the end site will reach, and ultimately the transcription will stop. Now this is DNA. These are the nucleotide base sequences. This is DNA, and RNA polymerase will bind on that point where the transcription is needed. This is the enzyme. This is RNA polymerase. This is a large enzyme which sits on the DNA at the point where the transcription is needed or it is to start. Now I told you that if we want to transcribe this strand, we will use this strand. So this strand is our guiding strand. We are using the code of this strand. So this will be the coding strand. And the other name of this strand is sense strand. And we are using its complementary strand to make our messenger RNA. So this strand is our template strand because we are using it as a template to make the messenger RNA. So this will be our coding strand of whose code we are using, and this is our template strand because we are using this as a template to make the messenger RNA. And another name for template strand is antisense. Now the transcription will start when this is the gene sequence that is to be copied. Now the transcription will start at the promoter site. This is promoter site or promoter sequence. You can say that it is the starting point, the start point or the start sequence. What happens is that there are various components of this enzyme. This RNA polymerase has various components of which few are involved in the transcription. First of all, here at the promoter site there is a factor. This factor is known as sigma factor. Now the process is initiated when this sigma factor is bound to this site. As soon as the sigma factor of this RNA polymerase binds to promoter site, the process begins. The promoter site is different in different cells. Promoter sequence of prokaryotes is different from promoter sequence of eukaryotes. Both are different. In prokaryotes there are 35, -35 and -10 sequences, and in eukaryotes there are -75 and -25 sequences. So the binding of RNA polymerase to the promoter is the first step in gene transcription. Now there is one subunit of this RNA polymerase which is known as the sigma factor. This factor is basically responsible for the start, and it also makes sure that the process is initiated correctly. This is responsible for correct initiation. Once the transcription process has started, the sigma factor is released. Okay, the sigma factor, when the transcription will start, the sigma factor will be released, and the whole transcription process will carried out till the end, where another factor will stop the process. Now here we have a sequence. This is the actual sequence of DNA. Now the newly synthesized messenger RNA which will be formed, we have to make sure that we are using a 3' to 5' strand instead of 5' to 3'. So our template strand will be 3' to 5' direction, and our newly synthesized messenger RNA which will be formed here will be in 5' to 3' direction, and it will have the subunits or nucleotide units attached accordingly. In RNA, we already know that in place of thymine we have uracil. In this way, the nucleotides are added to the newly growing strand of RNA. This is the second step in which the elongation of the RNA strand takes place. The DNA strands opens up, and at that site where enzyme is attached to the template strand, which will form this transcription bubble, and the transcription bubble will move down the DNA. As this messenger RNA is completed and it moves out of the DNA, the bubble moves forward till the point of termination. At the point of termination, there are stop sequences, and there is another factor present here. Here we have another factor. This factor is known as the row factor. Now as soon as the row factor is reached, the transcription process will stop. The stop sequence at the end of gene, it will terminate the synthesis of messenger RNA. The simplest stop signal is a series of GC base pairs followed by series of AT base pairs. The GC base pairs means that when the messenger RNA is formed, it will take this shape, which will